Hey guys, this is Miss Sato. I miss all of you. I hope that you are staying safe and healthy. Today's lesson, we're going to talk about DNA. So I'm going to go through your notes with you. So DNA, what is DNA? Well, we all did the packet. Hopefully we all did the packet. And we were able to look at different pieces of evidence that kind of helped you determine what DNA was, like what structure, what the bases were, what makes up the DNA structure, like a nucleotide. So let's go into the notes. So on slide 2, DNA is a nucleic acid. It's an organic compound. DNA stands for deoxyribose nucleic acid, a very long word. That's why we just call DNA DNA. DNA contains all the genetic information needed for the development and functioning of all living things. So the location of DNA makes up chromosomes, and chromosomes are an organized structure of DNA and protein. Chromosomes are found in the nucleus of a cell. Humans have 23 pairs and 46 in total, meaning that you get 23 from mom, 23 from dad, and that gives you 46. We're going to talk about more about what kind of makes you who you are when we discuss genetics. So the structure of DNA is made up of repeating nucleotides, which if you did the packet, you would have had to label something similar to what's on the slide. The circle is phosphate, next to it is a sugar, and then you have your base. So nucleotides are building blocks of DNA, and they're made up of 3 things: a sugar, a phosphate, and a base. So the sugar is the deoxyribose sugar. The phosphate and nitrogen bases, you have 4 bases. You have adenine, guanine, cytosine, and thymine. Yes, those might be hard words to pronounce, so you guys can just call them A, G, C, and T. The way I remember the structure of a nucleotide is that for me, the phosphate looks like a sun, the sugar looks like a house, and the base looks like a pool. Might not work for some, it might work for others, but that's what a nucleotide reminds me of. I guess I always have summer on the mind. So the shape of DNA, so DNA is a double helix. That was in your questions 1 and 2 when it asked you how many strands you saw in the images. So it's a double helix. It's 2 strands of DNA that twist around each other. We call it a twister ladder. They are attached at the nitrogen bases. Watson and Crick discovered the double helix, but with help from someone that's not really mentioned that often, which is Rosalind Frankel, who was in your packet. She actually took X-ray images of DNA and realized that it was repeating nucleotides or repeating bases, meaning that the shape of DNA had to be double-stranded and twisted. When we went to the DNA museum, you guys all got to actually see a replica, of what Watson and Crick designed in their lab. You guys were able to see the double helix structure in the museum, which I think is pretty cool that you guys were actually able to see that. So this diagram here just kind of shows you what the double helix looks like, what the bases are, how it's shaped, and down below you actually get to see what the nucleotide is. So you have the sugar phosphate backbone, and then you have the bases, like I said, guanine, cytosine, thymine, and adenine. So slide 7 just shows you the organization of this, meaning from like large to small. So if we start from the largest, we're going to start with the cell. Inside the cell, we find a nucleus. Inside the nucleus, we find chromosomes. Those chromosomes are made up of DNA, and made up of DNA are your genes. So the genes would be the smallest, and the largest would be your cell. If you look to the right, you get to see this cool diagram, which is a strand of double helix that's actually what we call replication. So this one strand is actually unzipping and forming 2 identical strands. We're going to talk about how DNA replicates later on, but today we're just going to focus on the structure of DNA, what it's made of, and how it looks. So the nitrogen bases, each nitrogen base has a complementary base pair. So adenine is always going to bind to thymine, and cytosine is always going to bind to guanine, meaning that A is going to bind with T and G is going to bind with C. Now, you can also have it the opposite way, meaning that you can have T bind with A and C bind with G, but they're always together. So I like to say, to remember, A goes with T and G goes to C at the Grand Canyon. You guys can make up your own term. Last year, my students made up add to group chat or add to Google Classroom, whichever helps you remember that A goes with T and G goes with C. But you can see here we have two strands, phosphate sugar base, and they repeat. This is just another diagram showing you that green is only going to go with red, purple is only going to go with yellow, meaning that A is always going to go with T and G is always going to go with C. So let's practice here. I did the first example for you. You see that you have strand 1 and you have strand 2. A is always going to bind with T. T is going to bind with A. G is going to bind with C. And C is going to bind with G. So I did the first one. I want you guys to also do this. This is good practice on writing complementary base pairs. So let's practice put the structures in size order from large to small. I did that for you in the previous slide when we had the cell. So I want you guys to figure out and write down from large to smallest. So genes and codons. Well, genes are a segment of DNA that codes for a protein or a trait. And codons are a group of 3 nitrogen bases that code for a specific protein. We call them triplets. And the code is the order of bases. So for example, you see here ATC, that's a group of 3, that's a codon. GGC, that's a group of 3, that's a codon. CCC, that's a group of 3, that's a codon. GAT is a group of 3, that's a codon. So each codon here is going to code for a specific amino acid that will actually code for a protein that codes for your traits. I know I just said a whole lot, but what it really means is that these codons are specific for your traits, meaning that you can have a group of 5 codons that code for a specific protein, giving you brown eyes, or you can have curly hair, or you can have straight hair, blue eyes. Do you have dimples? Can you curl your tongue? So when we get into genetics, which is my favorite, we are going to be able to talk about all what your genes do and what makes you who you are. The last slide of today's lesson, it's just showing you another large to small image. It's showing you a chromosome and 2 genes. So you have gene 1 and gene 2. And it just shows you how small the DNA is and how small your genes are. Along with this lesson, you guys are going to get a note sheet. I would hope that all of you do fill out the note sheet with the missing words. That's going to help you memorize all these different terms. It's a lot of new vocabulary word for the DNA unit. We just learned about a nucleotide. We just learned about the 4 bases. We just learned about what makes up a nucleotide, a phosphate, deoxyribose sugar, and a base. So I want you guys to kind of write in the notes. I miss all of you and I hope to see you soon. And I will speak to you guys soon. Bye.